Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Fitria binti Ahmad Munir and my matrix number is 23DBT 20F1036. Okay, today I want to explain about what is DNA extraction. So, what is mean by DNA extraction? DNA extraction is a method to purify DNA by using physical or chemical methods from a sample separating DNA from cell membranes, proteins, and other cellular components. Usually, DNA extraction is used to diagnose many medical conditions to genetically engineer both plants and animals. So, DNA extraction can also be used to gather evidence in a crime investigation. Next is the steps necessary for DNA extraction. So, for DNA extraction have three steps necessary, which are first, lysis, second, precipitation, and lastly, purification. In first step, which is lysis, the cell and the nucleus are broken open to release the DNA inside, and there are two ways to do this. First, mechanical disruption breaks open the cells. This can be done with a tissue homogenizer, which is like a small blender with a mortar and pestle, or by cutting the tissue into small pieces. Mechanical disruption is particularly important when using plant cells because they have a tough cell wall. Second, lysis uses detergents and enzymes such as proteinase K to free the DNA and dissolve cellular proteins. Okay, in step 2, which is precipitation, when you complete the lysis steps, the DNA has been freed from the nucleus, but it is now mixed with mashed up cell parts. Precipitation separates DNA from the cellular debris. First, sodium ions neutralize the next charges on the DNA molecules, which makes them more stable and less water-soluble. Next, alcohol, such as ethanol or isopropanol, is added and causes the DNA to precipitate out of the aqueous solution because it is not soluble in alcohol. And the last step, which is purification. So, in these steps, now that DNA has been separated from the aqueous phase, it can be rinsed with alcohol to remove any remaining unwanted material and cellular debris. So, at this point, the purified DNA is usually redissolved in water for easy handling and storage. So this is the DNA extraction biotechnics virtual lab. So start the lab. In this experiment, I need to isolate some DNA from a human test subject. This is the DNA. So press next to continue. This is the genetic testing, body identification, and analysis of forensic evidence. So, scientists isolate DNA for a variety of reasons, some of which includes first, genetic testing, second, body identification, and lastly, analysis of forensic evidence. Press next to continue. So, DNA extraction is typically the first step in a longer laboratory process. DNA extraction is an important part of that process because the DNA first needs to be purified away from proteins and other cellular contaminants. So, press next. This is the cells, and this is inside the nucleus have the chromosomes. Inside the chromosome has histone, DNA, so first I need to collect some cells from our test subject. So press next to continue. So this is the cheek cells. The skin on the inside of our mouth loses thousands of cells every day. These are the cells we're after. So press next to continue. The steps I will follow to purify DNA from a cheek swab are shown below. First, collect cheek cells. Second, burst cells open to release DNA. Third, separate DNA from proteins and debris. And lastly, isolate concentrated DNA. Press next to continue. This is the materials and equipment that will need to purify DNA from cheek cells. So these are the equipment and materials: warm water bath, microcentrifuge, buccal swab, micropipetters, lysis solution, sample tubes, concentrated solution, and others. So first, drag the swab over the inside of the test subject's mouth to collect a sample. So drag the swab over to the inside of the mouth. So this is the cheek cells. Next, I will place the swab into an Eppendorf tube. Look closely at the swab. Note that it is covered with hundreds of tiny cheek cells. Inside cheek cells is a nucleus, and inside the nucleus is DNA. So press next. So the end of the swab must be cut off, so I will be able to close the tube. Using the micropetters, add some lysis solution to the tube. Lysis is a Greek word that means to separate. So using the micropipettes, add some lysis solution to the tube. Okay. Then place the tube into the warm water bath. Okay. So the lysis solution just added contents to important ingredients such as detergent and an enzyme called proteinase K. The detergent disrupts the cell membrane and nuclear envelope, causing the cells to burst open and release their DNA. The DNA is still wrapped very tightly around proteins called histones, and the proteinase K cuts apart the histones to free the DNA. So this is the histone, and press next to the continue. So the cell have stayed in the warm water bath long enough for the DNA to be freed from the cells, and we have to remove the swab from the tube. Add some concentrated salt solution to your tube. So add some concentrated salt solution using the micropipette that have a tip. Okay. Add some concentrated salts. So the salt causes proteins and other cellular debris to clump together. Press next to continue. Place the tube into the centrifuge. In order to balance the centrifuge, a tube containing water is placed opposite your tube to become uh, the centrifuge balance. So click the lid to close the centrifuge and turn on. So inside the centrifuge, the tubes spin around at high speed. The heavy clumps of proteins and cellular debris to sink to the bottom of the tube, while the strands of DNA remain distributes through the liquid. So press next. So use the micropipetters to carefully remove the top liquid, which is contains DNA, and place it into a clean tube. The proteins and other cellular debris stay behind. So use the micropipetters to carefully remove the top liquid and transfer in the clean tube. Then add some isopropyl alcohol to the test tube. Okay, close. Then shake the tube. So inverting the tube several times, mix the isopropyl alcohol into your DNA solution because DNA is not soluble, which is does not stay dissolved in isopropyl alcohol. It comes out of solutions. So can see the clump DNA with your naked eyes. So this is the clump DNA. So press next. Place the tube into the centrifuge and click on the lid to close it and turn it on. This time after the sample spin in the centrifuge, the DNA sinks to the bottom of the tube. So now place the tube into the centrifuge and close the lid, turn on. Okay, now it's done. So once the liquid is removed and the DNA is allowed to dry, you can redissolve it in the solution of your choice. You can store it in the freezer for many years, or you can move on to your next experiment. You have just extracted DNA. So it's done.